So with that, I'd like to introduce Seth Crawford, who is one of the co-founders of Oregon CBD. Oregon CBD is one of the larger and well-regarded genetic hemp genetic providers, seed breeders in the state, and they've been working on some new technology, these triploid hemp seeds or hemp genetics that hopefully will reduce the potential for seeded crops. And so we're sort of thinking about like the seedless watermelon. How do we like end up with that fruit that's able to grow and produce without having viable seeds in it? And so with that, I'd like to turn it over to Seth and just have him tell us what they've been working on.
Fantastic. And I'm going to try to share my screen here, Gordon. If that's all right.
Please do.
All right. Awesome. Is that coming through?
That looks great.
Okay. Yeah, I really appreciate the invite and you putting this on and OSU sponsoring it. It's a fantastic resource for farmers, and really excited to be able to share this information with y'all. I'm going to do my best to pack about 3 years of research into 15 minutes, but definitely stick around afterwards. We'd love to be able to answer some questions. It's really where the magic happens. So I'll say from the outset here today that the journal article detailing today's information is in preparation for publication and should be available later this spring. We'll have that up on our website when it finishes the review process. So with that said, I'm Seth Crawford. My brother and I co-founded Oregon CBD in 2015. We're the state's oldest continually operating hemp farm and the nation's leading supplier of feminized cannabis seed. Farmers around the country have used our seeds to win over 40 different awards in trim flower competitions. I noticed that Paul Murdoch was on here. Hey, Paul. Paul's farm beat out all other varieties this past year, including THC dominant varieties, to win a flagship award at the Cultivation Classic. So hearty congratulations to him and his team. Other varieties that we have, several of our cultivars, Lifter and White CBG in particular, are now being used as the benchmarks for university field trials across the country when they're assessing new varieties. We have a 49-acre research campus located in Independence up here in the Willamette Valley, and it's home to 40 cannabis scientists, chemists, and other horticultural professionals. And we're operating one of the most advanced, if we can say so ourselves, cannabis breeding programs in the world at our little farm, and we're collaborating with other scientists to better understand this incredible plant. So today I'm going to talk a little bit about seedless triploids. And these, in short, are cannabis plants that are incredibly resistant to pollination, as well as other exciting attributes brought on by extra genome copies. So our development of triploids was born out of the trauma of losing several crops to cross-pollination events in 2016 and 2018, and honestly hearing horror stories from growers year after year. If you've grown cannabis, you likely understand how devastating pollen drift can be on your crop, and that it can travel hundreds of miles in the right conditions. Fully pollinated plants produce about 50% less flower weight and about 30% less cannabinoids. So a seeded crop is essentially worthless if you're growing for trim flowers, and still devastating if your end goal is biomass and extraction. As we've also seen in the first 6 years of production in Oregon, feral populations of cannabis are regularly establishing themselves in fallow fields and have become a big enough concern that ODA is now working on rules and penalties for landowners who fail to control rogue plants. Having a plant incapable of being pollinated fixes a lot of those issues and threats and lets farmers put their energy into growing great crops instead of worrying. We've been working hard to get the results of this truly remarkable breeding advancement into the hands of farmers in time for planting this year. So to start off, to understand triploids, we need to establish a little bit of terminology. So chromosomes are packed together collections of DNA, which is the underlying genetic instructions for life. It's helpful to think of chromosomes as books of code, which make up a library that is an organism. Every species has a unique number of books in its library. Humans have 46, naked mole rats, for example, have 60, and cannabis has 20 in the diploid state. So this library size is referred to as the chromosome set number. Ploidy refers to the number of homologous sets of chromosomes in an organism. Organisms with a single set are referred to as haploid, 2 sets are diploid, 3 sets are triploid, 4 sets are tetraploid, so on and so forth as you go ever higher in those ploidies. Any organism with a ploidy higher than 2 is referred to as a polyploid. In sexually reproducing species like cannabis, 1/2 of the chromosome sets are inherited from each respective parent. For humans, this means our individual genome is actually a fusion of 2 different genomes cut in half during reproduction. We're truly 1/2 of both of our parents. The same is true in sexually reproducing diploid plants. So while most vertebrates are diploid, many plants are actually polyploid. And these include major agricultural crops of critical importance to humans, such as coffee, bananas, strawberries, bread wheat, oats, citrus, potatoes, and tobacco, to just name a few. The main point to understand, I think, is that polyploidization, this process of increasing your genome size, is a natural phenomenon that has occurred many times in nature and has led to the creation of the ancestors of many modern crops. Polyploid induction in nature tends to correspond with extreme climate events or natural disasters and lead to increased diversity within a species. The only documented naturally occurring tetraploid cannabis population, so this would be a polyploid cannabis population, was found at 14,000 feet in the Himalayas by an Indian research effort about 10 years ago. We can sort of assume that the extreme climate and altitude may have been the causal agent responsible for that particular event and partial speciation. We can think of this as nature's way of creating options for diverse responses under changing circumstances. It's also important to note that these events are not changing the underlying DNA or introducing any new genes. They're simply whole genome duplications of what was already present in the organism. In the case of polyploids, more gene copies tends to confer better adaptability and resilience. Plant breeders became aware of the remarkable superiority of polyploids in the mid-20th century and have induced polyploidy and or used natural polyploids in many ways to obtain increasingly improved plant cultivars. Some of the most important consequences of polyploidy for plant breeding are the increased size of plant organs. So we're talking about bigger flowers, bigger leaves, bigger stems. The buffering of deleterious mutations, increased heterozygosity, and superior heterosis or hybrid vigor. Cultivars have been generated with higher yields, improving the product quality and increasing tolerance to both biotic and abiotic stresses. Polyploidy is basically a tried and true method of improvement in many crops. And so now that cannabis is legal, we have the opportunity to explore the effects of polyploidy in this particular species. An important bonus, and obviously the whole point of this talk, is that plants with odd chromosome set numbers, so we're talking about triploids with 3, pentaploids with 5, and so on, often prove to be infertile. So this is what makes seedless watermelons, bananas, hops, and now cannabis a reality. So the first comprehensive chromosome scale cannabis genome was published just recently. This is 2020. There's several other lower quality assemblies publicly available in NCBI, but the CS10 assembly that's pictured here is currently considered the reference. And this tells us that the cannabis genome size is approximately 800 megabases in size as a haploid and 1.6 gigabases as a diploid. And that the plants we've all historically grown as diploids contain about 30,000 total genes. And there's about 72% of the total contents of that genome that exist as non-coding repeat elements. So for example, cannabis's closest relative, hops, is about twice the size, as a genome that's twice the size, and it has similar levels of non-coding repeats. So that's a really important point for researchers to know as you're starting the process of identifying potential polyploids. You really need to know how big the actual underlying diploid genome is. So polyploids at Oregon CBD. Our polyploid development program was designed and led by Dr. Shen Chen, who's a OSU PhD, who is now an assistant professor at NC State. Congratulations to him. Awesome guy, beautiful program, has done some amazing work. Definitely a leader in the industry. Our goal originally was to convert our diploid breeding stock that we've been using and that we develop on a regular basis in new forms into tetraploids and potentially beyond, depending on how far we can take ploidy manipulation in this crop. We were also aiming to test the fertility of various ploidies in controlled environments. So this is the does it get seeded, and how often do you find seeds in different ploidy crosses, as well as trying to test the agronomic traits of various ploidies via controlled crosses in initial field trials. So controlled genome doubling events require your initial plants, cell division inhibitor, steady hands, patience, and a flow cytometer, which is a really cool piece of equipment capable of measuring an organism's cell size. So to do this, you take hundreds of asexually propagated clones of a target plant genotype and prune them back until a single growth point remains. And then in our case, colchicine is applied. Most of the plants die within a month, but there are a few survivors, and they get screened by flow cytometry. And any confirmed tetraploids move on to the stabilization process, which takes about 8 months from the initial treatment. At each step in this process, plants are rescreened over and over, as they have a tendency to revert to lower or mixed ploidies until fixed. If a plant has 8 shoots, all 8 shoots are tested. And this continues until stability is attained. And then the plants are ready for seed production, but not before a final flow screening before those plants go into our production greenhouses. Tetraploid germplasm is always maintained in a separate greenhouse to avoid mix-ups. And at this point, we've now successfully created tetraploid versions of all of our classic CBD and CBG varieties, as well as several of our new CBDV production mothers and type 1 plants that we have in our germplasm library, and have now incorporated this streamlined conversion process into our breeding program. But the moral of the story here is that it takes time. It takes us about 8 months from the start of the process. And to get to the start of that process, you had to do basically 3 years of previous breeding to get a plant that's worthwhile of locking in those traits. You can see in these images the resulting peaks generated by the flow cytometer when testing plant material of various ploidies, as well as microscope pictures of their actual chromosomes. This is really cool. This is one of the first publications that's going to actually show cannabis chromosomes. Using multiple methods to validate ploidy levels is especially necessary when creating baselines for further analysis. But the chromosome pictures help to convey the cellular changes present in polyploids. There's really a lot more genetic information in these plants, and you can see it when you get close enough, in this case with a very powerful microscope. Diploid cannabis plants contain about 60,000 genes total and have a genome size of 1.6 megabases. Triploids contain 90,000 genes and have a genome size of 2.4 gigabases. Tetraploids contain 120,000 genes and have a genome size of 3.2 gigabases, which is roughly half the size of humans or about the same size as hops. These extra gene copies are what most scientists who are studying and plant breeders who are studying this believe drives the resilience and adaptability of polyploids compared to their diploid relatives. So seedlessness and triploid cannabis, yeah, man, that could be a wonderful thing. But we really had to design some experiments that captured that quality and allowed us to assess the results. So to do this, we created 6 crossing combinations to assess seed production and viability. Diploid, triploid, and tetraploid versions of the same F1 hybrid cross were pollinated by diploid and tetraploid pollen donors. These trials were conducted in sealed greenhouses, and the target plants were subjected to literally massive amounts of pollen. This was intended to simulate a flower crop being grown side by side with a 100-acre grain farm. So the results here show that diploids pollinated by diploids, this is your normal cannabis cross, produce about 2 times as many seeds as tetraploids pollinated by other tetraploids. This suggests that ploidy increases actually result in lower fertility in cannabis as you move up the ploidy ranks. Tetraploids are not viable as pollen donors for diploids, but they are able to induce many problematically empty seeds. The key finding is that seed production is essentially non-existent in triploids when pollinated by either a diploid or a tetraploid. Even under substantial duress, they remain nearly seed-free with a maximum average. Remember, this is a sealed greenhouse with just pollen everywhere. And in that condition, we saw a maximum average of 6 seeds per plant, per triploid plant, when pollinated by diploids, which has the most viable pollen, in a controlled pollen inundation zone. Triploid pollen donors are not reported in our work that is up for publication because they're sterile and they don't produce viable pollen, which is a really important point if you're a farmer and you are trying to protect both yourself and your neighbors. On the agronomic side, in addition to relative seedlessness, triploids are also superior in many agronomic traits relative to their diploid counterparts. We conducted outdoor field trials at our campus here in Independence in 2020 with several different diploid, triploid, and tetraploid crossing combinations. What we found was that flower production increased anywhere between 30% to 100% over the diploid controls, and that was variety dependent. We really saw the largest gains seen in day neutral varieties, so auto flowering varieties. The yield differences are statistically significant, but honestly, the visual difference in plant size, the larger leaves, the larger flowers, the thicker stems, denser trichome coverage, that tells you all you need to know about triploid vigor. There's a slight increase in total cannabinoid content in flowers and a marked increase in trichome density on sugar leaves near those flowers. Additionally, olfactory compounds, so the compounds that make cannabis smell, were noticeably more abundant in the triploids and tetraploids, frankly, compared to the diploids. But we didn't actually test these for total terpene content in this particular study because this is going to be something that's coming out in the next few years. But terpenes really tell less than half the story, and most of the major aroma components of our varieties are not actually terpenes at all. So we forewent that. This year, we have field trials across the US scheduled this summer with about a dozen different universities to assess, in particular, the pest and pathogen response differences between ploidies in diverse microclimates. But the early results from our internal trials in Oregon last summer suggest that triploids are much more resilient to any challenge they face relative to diploid counterparts. There's not much of a surprise that our findings are similar to what's been reported in the literature for polyploid cannabis over the past year, as there has been significant commercial and academic interest in them for obvious reasons. But ours was the first to really do this in an outdoor environment, and then also to subject them to that pollen to demonstrate that they were seedless. We've got some additional collaborations going on at OSU. This is sort of the bookend to our polyploid development work. We've got continued support of several funded research programs at OSU, and it includes the labs of Kelly Vining, Pat Hayes, and Ryan Contreras. Research is underway, including ploidy induction optimization protocols in cannabinoid-free germplasm. So these are plants that do not produce any cannabinoids, but still produce olfactory compounds. Assessing chromosomal stability in induced polyploids over time, genome reduction techniques, tissue culture media assessments of diverse genotypes, RNA sequencing to identify sources of THCA variation in our CBG varieties, and probably most importantly, the construction of the first cannabis pangenome derived from our collection of whole genome data, which is the largest in the world. The pangenome work is really exciting as it's going to be a constantly evolving tool for future generations of breeders to work with and benefit from. And this work is currently supporting 3 master's students and 2 postdocs. And we're really excited to continue working with OSU to push the boundaries forward in cannabis. So we're really excited to be able to offer farmers this evolutionary leap, truly, in cannabis breeding. Triploids are the future for cannabinoid production. They virtually eliminate the risk of a seeded crop. They protect yourself and your neighbors from errant pollen. They substantially outproduce diploids and help protect Oregon's unique production environment from the spoilage associated with feral hemp populations. Our company is releasing nine different triploid varieties for the 2021 production season, which include CBD dominant varieties, CBG dominant varieties, CBDV dominant chemotypes, and we have some photoperiod sensitive and day neutral traits included in those as well. And the best part is for farmers in terms of value added, these are all certified organic by the Oregon Tilth and non-GMO by the NSF. So as awesome as these 2021 varieties are and as excited as we are, we're really looking forward to the new flavors, new cannabinoids, new flowering response traits, and new ploidy levels to come over the next few years. I'm really proud of our entire team for making this possible. There's obviously a lot that goes into creating quality seeds at every step in the process, from the foundational science to the final seed production to shipping and on-farm advice. And we really truly have one of the best teams in the business. We're always grateful for our farmers as well. They do a great job of making us look good year after year, and we can't thank you enough for that. We love sharing the latest and greatest, especially when it translates to even more successful clients. So for more information on this, please feel free to visit our website or reach out to either myself for technical questions about ploidy, or our farm relations manager, Imre Garcia, for ordering information. Imre's based in Grants Pass. He's in Southern Oregon, where we all grew up, and he's a wealth of knowledge and information. So with that, I'd like to thank you all for your patience, and I can take questions now, later, or whenever. Happy to hang out.
Awesome. Thank you, Seth. I really appreciate that. Just doing something that's sort of different, that I guess I feel like as a university employee, they ask us not to like endorse any one product or another. And that the Crawford brothers are great supporters of us at the university. We've like not tried out these triploid varieties independently, like across the state. And the reason and there are lots of like good seed companies. I think that you all are the only ones making triploid seeds. Is that right?
That is correct. Yeah.
And that's really why you get an invitation here, because you're doing something different that I can, there'll be more to be learned about this as it progresses, but clearly are some benefits. I know as like an agricultural scientist that the manipulation of ploidy is something we've done or like utilized for centuries, and has a range of benefits that really fall in line with what we see here in terms of plant characteristics. And so makes some sense. I really appreciate that information that you shared. Let's just do like one or two questions out of the chat box here. And if you're willing to stick around, we'll do some more at the end. Question on this like triploid process versus our like 0.3% THC limit. You said a slight increase in cannabinoids. Is that across the board?
Yeah, it is. And the main reason for this is that, and this might not be known by everybody on the call, but THC in CBD dominant plants actually comes from the CBDAS synthase. The CBDAS gene that produces CBD also produces THC, CBC, and 5 other compounds that still haven't been characterized yet. So basically what we're doing is creating more copies of that same gene, which means that you're going to have basically more expression of those compounds. Not what we found is it's not statistically significant in terms of total cannabinoid content. I think on a per plant basis, there's different ways of operationalizing the test. What we're seeing now in our production environment is that tetraploids there's just more trichome coverage. So it stands to, for us to believe that there's probably an increase overall. To go back to that question, the overall ratio of CBD to THC does not change in these plants. And that's because it's a fixed component of the actual gene itself. You just have more copies of that gene now. In our CBG varieties, those are all fully compliant due to an inactivated THCAS synthase. And I've got, if anybody's interested in that, there's more information with a paper that we just put out about that particular gene that we use to make our CBG varieties. And then we've got these new CBDV dominant plants that produce half the total amount of THC that traditional CBD crops do. So obviously the science is definitely ahead of the legislation in that regard. There's obviously talk about a 1% total THC cap, and that's scientifically accurate for type 3 CBD producing plants. But there are other compounds and other ways of going about this that allow you to create a product that's actually fully compliant even at harvest. It just means that you're producing CBDV or producing CBG or other compounds.
Awesome, thank you. And then I guess one more question that's just on my mind. These are still feminized seeds that you could have triploid males, right? Is that right?
No, there's so in these plants, and we're seeing the same sort of outcome that we've seen in other. So in our feminized diploid seed, about 1 in 4,000 plants has a male phenotype, even though it's genetically 100% female. It has 2X chromosomes. The same thing is true in these plants, although the rate is a little bit lower. The added benefit of this is that the triploids don't produce viable pollen. So even if you do have one of these 1 in 4,000 plants that pop up, it's not going to pollinate your neighbor just due to incompatibility with diploids and tetraploids, along with your triploids. So this is a great way of minimizing any types of pollen contamination issues. And just to step back, because I think there was the idea behind this is, well, how do you make these triploids? And this last slide really highlights it. To make a triploid, you have to have a tetraploid and you have to cross it with a diploid. So you end up with 2 contributions from your tetraploid parent and 1 contribution from your diploid parent. And that gives you 3 sets of chromosomes, 3 homologous sets of chromosomes, making it a polyploid and infertile.
Awesome. I appreciate that information. And yeah, if you'd be willing to stick around for a little while longer, maybe